I'm from the TATAA BioCentre, and we've been working in the qPCR field for 25 years now, since the very beginning in the end of 1980s. Essentially what we do is that we do training courses in all aspects of molecular diagnostics, from experimental design, preparation of the samples, the actual analysis, and data analysis as well. We do offer a lot of services, particularly to pharma, biotech companies, analyzing their data, and we are also a core facility. And in particular, we've been very interested in quality control and quality aspects of molecular diagnostics in general. And there are many reasons why one should be interested in that, and usually I try to give some example from real life, and I think this is a pretty interesting example. I'm from Europe, so this is from Germany. And I don't know if you caught that in the essentially in the news. This is something that happened a little more than 5 years ago, when the whole entire Germany was essentially chasing a serial killer, because all the traces they found at the crime scenes, they shared a typical DNA signature, and actually it turned out to be a female. So the whole Germany was chasing this female. And obviously this female had been active killing people for almost 10 years. So this is really exciting. 40 crime scenes. Wow. Could make the Guinness Book of Records. But what it actually turned out to was that it was the cotton swabs that were used that were contaminated, because that particular woman was working in the production of the cotton swabs. And of course, the process was not certified. So there can be quite important impacts of not, working under quality guidance. So what we did already now about 5 years ago, together with some colleague of ours, opinion leaders in the qPCR field, well, we noted there was a lot of mistakes being done in molecular diagnostics, and in particular in real-time PCR. So we put together guidelines called the MIQE Guidelines on how you actually should report your data when you want to publish something. It doesn't really tell you that you have to achieve certain fulfillments, but rather how you have to report it in order for other people to be able to judge your results and to be able to repeat your experiments. Today, at least 15 to 20 journals are actually requiring that the manuscripts are submitted according to the MIQE guidelines, and we do see quite some impact on that. However, as the qPCR field is maturing and becoming more and more routine-based technique, we need also guidelines for routine diagnostics. So in Europe during 2008 and 2013, we had a major program in establishing guidelines how to work in real-time PCR. A total of 16 partners were working on that. It was coordinated by QIAGEN, and one of the partners was SEN, which is the European Standardizing Organization. And essentially, we did a number of things. Among those were so-called proficiency ring trials. So we took a sample that was distributed across routine laboratories in Europe. Some 200 laboratories participated, and it could be like just a regular blood sample. And we asked these routine laboratories to use their standard operating procedure, extract the RNA, and send it back to us, and we tested the quality. You know what? 30% of European routine laboratories have issues purifying intact RNA from a blood sample. This is what you learn from these kind of ring trials. And that's, of course, very, very serious. So what happened was that the goal of this project was essentially, first of all, to develop new technologies and approaches, how to secure high-quality samples, and also to generate data that could eventually become standards for the use in routine. So the first thing that happened after the SPEEDIA project was that SEN, the European organization, initiated work to set up standards for the pre-analytical process. That was in 2010. And end of this year, they will become public, and they will start to be enforced in different countries in Europe. And last year, a group was formed at the global level within ISO to generate 8 new guidelines on the pre-analytical process. So the groups working on it was in CEN, that's a European organization. It's the TC 140. TC, that's the technical committee. In ISO, it's TC 212. For those that are interested, I'm a member of those committees or those groups, and I'd be happy to discuss this more with you. The particular guidelines are being developed. That is the pre-processing of RNA and protein from snapped frozen tissue, DNA, RNA, and proteins from FFP tissues, and RNA, DNA, and circulating cell-free DNA from blood. And if you wonder whether you are affected by this or not, well, these are the countries that are essentially applying or adopting these ISO guidelines that are being developed. So, and what about the techniques that came out and the IDs we got? Well, particularly when analyzing RNA, a major issue is RNA integrity, because RNA that is kept in room temperature or under, or perhaps is preserved by chemical fixation, undergoes degradation. And we know that, and we typically do some QC using, for example, capillary electrophoresis, or using one of the automatic systems like the Biorad or the Agilent Bioanalyzer or Experian. This typically works pretty well, particularly on samples that have fairly high quality. But if you look at, for example, degradation over time at room temperature of a piece of liver or either powder, you see that the so-called RQI, this is a Biorad instrument, the RNA quality index, or the RIN number in the bioanalyzer, RNA integrity number, decreases. However, you see plateaus. What does that mean? Does it mean that suddenly we don't have any more RNA degradation? Not really. But what we see here is that these instruments are really not sensitive anymore to further degradation. And usually it is in this area where it becomes critical to know whether there is really something there that we can analyze or not. And furthermore, when you do this kind of analysis, you're actually analyzing primarily the 28 and 18S RNAs, that's ribosomal RNA. It's not mRNA that you're typically interested in. If you do specific degradation studies, these are time courses of a model system, it's oocytes, and we monitor RNA degradation. You actually see that 18S RNA is so much more stable than most of the mRNAs there. So it doesn't really reflect the integrity of the mRNA. So the question is, are there better methods to measure the integrity of the mRNAs that we are really interested in? A couple of years ago, something called the 5' 3' assay was proposed, and initially by Tanya Nolan, Steve Bustin. It was made available by Sigma for a period of time. And the idea was that if we transcribe or reverse transcribe mRNA using an oligo-DT primer, we will get specific transcription from the A tail, and if we then analyze the 5' end and the 3' end of the same mRNA, if the RNA is intact, there will not be a bias, but if the RNA is degraded, there will be a bias because we will not get full-length transcripts. And indeed, this 3', 5' assay works very, very well in model systems. This is an intact RNA. This is a degraded RNA. You see clear shift between the 3' and 5' assay reflecting the partial degradation. However, in practice, it does not work. There are many, not only us, has tried that. In fact, spent several years trying to get this working. No, you don't really see a preference in this assay for neither of the ends really, and this is just like real samples, liver samples, time courses, and there's really no difference between the ends. The reason for that, it's not fully clear, but we suspect that in a degraded sample there's so many short fragments around, so we don't get the specific priming at the A tail anymore, and that's really the main reason it does not work. So with Inspedia, what we did was that we did screen for a large number of markers, and we actually managed to identify a marker, an endogenous marker, that is virtually resistant to RNase digestion. It's not an mRNA. So that's probably the reason. So what we have in this graph is it's a titration with RNase. So we added RNase to it. This is the RQI value, so you see it goes down. This is the CQ value of a regular mRNA, and this is essentially the CQ value of the resistant endogenous marker. And that's very useful because we can now create a delta CQ between the stability marker and reference gene, and we can easily monitor RNA degradation, and you can see it follows very well the RQI, the integrity index, but furthermore, it does not plateau. You can monitor the degradation further down. Then, if you also have other types of degradation, not only RNases there, but chemical degradation, because you're working with FFPE material, the RNA will be degraded chemically. And then the way you can approach that is to amplify the mRNA, or rather the cDNA, using amplicons of two different lengths, a shorter and a longer amplicon. And the logic here is, of course, again, if the cDNA is intact, you will get the same yields of the short and long amplicon. However, if the RNA, and then of course the cDNA is shorter, then you will get a higher yield for the short amplicon. And indeed, this is in fact what you see. This is just RNA that is heated. And you clearly see that you have degradation, of course, reflected by higher CQ of both fragments, but there is a larger dependence for the longer fragment. And again, if you form the delta CQ, you can clearly monitor the degradation of the mRNA. And these signals are large. This is a delta CQ going from 0 to 7. It's very easy to measure with very, very high accuracy. So, and this also reflects heat degradation, chemical degradation like FFP material, but also UV degradation, which is actually not sensed by the capillary electrophoresis. It's not sensitive to UV degradation. And the last thing that came out of SPEDIA is something we call valid prime. And this is essentially a assay to test for genomic DNA. And why is that important? You see, many of you are doing gene expression profiling. And when you do gene expression profiling, you have to be concerned that the primers you design for your transcripts may actually amplify the genomic DNA as well, if there is some genomic DNA background. Many companies teach that if you design your primer spanning an intron, you will not amplify the genomic copy. And the reason is the two primers will be far apart on the genomic level, and therefore there will not be time enough to actually amplify it. That is true, but one needs to know that actually 15% of human genes, they actually have pseudogenes. And about 50% of the pseudogenes actually lack introns because they are not copies of the original gene, they are copies of the transcript. And all those pseudogenes will be amplified even with intron spanning primers. And a gene can have multiple pseudogenes. GAPDH has over 60 pseudogenes in human and over 300 in mice. So the background can be substantial. So typically, in the old days, one would run RT negative controls. You would run a parallel reaction, leaving out the reverse transcriptase, and that would be your control for the genomic DNA background. However, most labs don't want to do it because it gets very, very expensive. So what we did was that we designed an assay. We call it valid prime, and it's an assay that is specific for genomic DNA, but it's targeting a non-transcribed locus, which means that it's not going to amplify cDNA. And then all the samples are assayed with valid prime. So essentially, this assay is measuring the genomic DNA background, and the sequence targeted is present in exactly one copy per haploid genome, and is highly conserved. This is not sufficient for correction. But if also the assays are tested on a reference genomic DNA, we know the sensitivity of those assays for genomic DNA, and from that combined information, we can easily correct for the genomic DNA background. And you see that if you analyze, let's say, 4 genes and 5 samples, it's instead of doing 4 times 5 RT negative controls, you do 4 plus 5 plus 1. And in larger studies, you can save 95% of your reagent costs. Furthermore, the reference genomic DNA is being calibrated against the NIST standard reference material 2372, which is a human genomic DNA standard, calibrated by digital PCR, so we can actually tell exactly how much genomic DNA there is there. And that information will be important for the next step where we'll be actually evaluating the performance of an instrument. So how do we test the performance? Well, typically when we test the performance of a workflow, a test, we would make a standard curve. We would also determine the limit of quantification and the limit of detection. How is that done? Well, this is a typical standard curve. We have seen those before, and typically from a standard curve, the PCR efficiency is determined, including the confidence interval. In fact, the confidence interval turns out to be a very important parameter. The limit of detection. What determines the limit of detection? Well, in most cases, everything is working perfect. It's actually the sampling ambiguity. The fact that if you collect an aliquot of a larger container, you may occasionally get a certain number of molecules, and next time you get a different number of molecules. Let's say that the container contains an average of 1 molecule per mil, and you sample 1 mil. Well, from the Poisson distribution, and this particular case was 1 molecule per mil in average is shown in pink, we see that the probability is 37% we actually get 1 molecule in that sampling. It's 18% probably we get 2 molecules, and 7% we get 3. But there is a risk of 37% that the sampling is actually negative. And from those probabilities to get a negative sampling, we can calculate the theoretical limit of detection. If we work at 95% confidence, we can enter this graph here, and we actually read out that the theoretical limit of detection of actually any experiment where you have a sampling ambiguity, not only qPCR. Digital PCR is the same thing. NGS is the same thing. Nanostring is the same thing. The theoretical limit of detection is 3 molecules due to the sampling ambiguity. You can't go below that if you have a sampling. What about a limit of quantification? Our ability to quantify can be determined from the variation of replicates. You can see that as you go down in concentration, the spread of replicates increases. And then you have to define the threshold. And the threshold really depends on the test system, but it's quite common that you would set the threshold of the quantification around a CV, or a residual standard deviation, of 35% of the back-calculated concentration. So let's see how that works on a real system. So the system we have been testing is the IntelliQube from Douglas Scientific. There's a picture here, but there's a real one next door, so I really think you should go there and have a look at it. It has a fairly high throughput because it has 768 well array tape. That's twice the size of the 384 well plates. And it has like an intermediate also reaction volume. It's smaller than conventional qPCR, but it's not as low as digital PCR, and that actually has a number of advantages. And it's a fully integrated system with liquid handling, detection, amplification, and data analysis. So let's see how that works on real data. So I will take the data more or less directly from the instrument. So I use a wizard. I identify the Douglas Scientific IntelliQube here. I will select the data file, and this is the data as they come from the instrument. And indeed, this is very common that we have missing data, as we heard in the previous presentation. However, modern software for analysis will handle that actually automatically. So these are just the data. Here are all our samples. There are a large number of samples because it's 768 reaction wells altogether. Here are the concentrations for the standard curve, and here are replicates. I don't need to do any pre-processing. I just load the data. There are missing data there, I know that. And I will start doing just a standard curve analysis. And there are some outliers here. We'll come back to that. But I just wanted to show you how these data look. So this is typically how data look on a standard curve, where you have gone from pretty large number of molecules down to a single copy. This is very typical because what you see, there's a large number of replicates here, and the spread of replicates increases as you go down to lower number of molecules. And this is expected. It's not a malperforming instrument. What we see here, that's a sampling ambiguity, because the smaller number of sample molecules we have per sampling, the larger is the variation across the samplings. Before analyzing really the standard curve, we have to make sure we're on the dynamic range. So what we do next is that we actually do the limit of detection analysis. So we do the limit of detection. We don't want to skip anything, so we just press run. And at the limit of detection analysis, we essentially register the number of positive samplings we have as a function of concentration of the molecules we load. And you see that, of course, at higher concentrations, we have only positive samplings. At lower concentrations, we have smaller numbers of positive samplings. Here we loaded an average of 1 molecule. And by fitting the data, we determine the limit of detection. And you can see it's 2.95. That's essentially the theoretical limit, which means that the instrument is actually detecting anything we put in there, and the limit of detection is only limited by the sampling ambiguity, which is due to the Poisson distribution. We can't do anything about that. What about our ability to quantify? So let's go and do the limit of quantification analysis. That's shown here. So what the software now is doing is that it is analyzing the spread of the replicates, and it is calculating the coefficient of variation as a function of the concentration. And if we, in our lab, as we typically do, have a threshold at 35%, that is, in order for us to report a quantitative result to our client, the precision has to be within 35%. So the limit of quantification will be the lowest concentration that gives you an accuracy that is 35% or better. And for this particular system, it's 32 molecules. Okay? So having done that, let's go back to the standard curve. But then what we should do in the standard curve is that we have to actually remove all the concentration below 32 molecules, because they are outside our quantitative range. So what I do, I just create groups of the different concentrations, and then I will remove all samples that had a single molecule, all samples that had only 2 molecules per in average, four molecules in average, 8 molecules in average, and 16 molecules in average. That means that what remains are samples that had at least 32 molecules, which is above our limit of quantification. And now I can make a standard curve again. And it's here. So now you see, we don't see the increase in spread anymore, and that's the reason is that we are within the dynamic range. We don't have a problem with sampling ambiguity, and there are no outliers anymore because we don't have outliers in this range. And if we look at the report, we also, there's a lot of data here. So let's look at the PCR efficiency that is calculated here. The PCR efficiency is 99.6%. The confidence interval is 97.7 to 101%. So essentially, very hard to get something working better than this. So the last thing I will show on this instrument, validating this instrument, is, I mean, we've heard so much about that, but the digital PCR. So we take a single sample and we essentially spread that across the entire plate with 768 wells. And we do the amplification. So let's see how it works on the IntelliQube. By the way, these are the guidelines for digital PCR that we published a few years ago, those that are interested. And what we did here, loading an average of 2 copies per well, we could then, performing essentially the PCR, we could, from the number of positive wells, do back calculation and calculate how many copies per well there actually was, and we end up with 1.96. And I should say this is the analysis with a valid prime assay on the genomic DNA calibrated against the NIST standard. That's why we know the number of copies that we are actually loading. We didn't get as good with when loading a single copy per well. We don't really know why. Maybe there are small losses with a single copy, but probably in the dispensing. We're not sure about that yet. But with 2 copies per reaction, we are essentially picking up everything. And so in conclusion, we evaluated the IntelliQube using the valid prime assay and the calibrated genomic DNA from NIST. We got a PCR efficiency, 97.7 to 101.7%. Best estimate is 99.7%. Limit of detection, that 3 molecules, that's the theoretical limit. It's not possible to go below that. Limit of quantification at a CV of 35% with 32 molecules. And sensitivity per well is 1 to 2 molecules. But remember, we're talking about larger volumes here than in conventional digital PCR. It's very, very important. And so we have a PCR recovery close to 100%. And just as a last slide, together with Douglas Scientific, there's more information there. Booth, we now have an offering that if anybody's, or those that are interested, can contact Douglas about an opportunity to get a free profiling experiment on the IntelliQube. We will provide, assuming we have those assays, we will provide grand performance assays. That's assays that have been validated down to single cell work. So we have a library of assays down to single cell profiling. So feel free to contact Douglas Scientific in their booth, number 2 here, and you can join this opportunity. Thank you very much for your attention. I'll be happy to take your questions.
Yeah, I had a couple of related questions about this RNA degradation resistant molecule. Is this something that's published and or commercially available?
It will be. It's being written up, so it will be published probably in a few months' time in BDQ, biomolecular detection and quantification. It will also end up as a product. We do offer it already, and we do that. We use it in services for our clients, but it's not on the shelf yet. But it will, because it really, really works very well. We use it because it fits the workflow. It's much easier for the workflow.
And this is some endogenous human RNA.
It's not an mRNA. That's the thing. It's an endogenous RNA.
So I guess my related question is, since most RNA degradation is enzymatic, and there are a variety of different RNases that are transcribed at different levels in different people, what validation have you done to establish that it is resistant to degradation? I mean, what experimental?
Actually, just a comment. What we have actually found is that there is even more issues with FFP material, which have even more degradation. That's not enzymatic. But what we have done is a number of model systems. So the model systems are essentially tissue, powder, and we have the third model system we used were oocytes, like from amphibian oocytes, that you essentially freeze thaw, so you make you let the RNases free, and it will degrade the RNA. And that's actually the traces that we've shown. We have also a model system that we just added RNAs to it, but 3 of the systems I showed there were actually biological materials.
Any more questions?
Here in the front.
How do you account for that in your calculation for LOD?
You know what? First of all, we don't get positive LODs. You don't get positive NTCs when you run. This is a very good point. But when you run conventional qPCR, you don't. And the reason is that you are actually monitoring the entire response curves. So it's much easier to separate a false positive from a true positive, because in most digital PCR you only have the endpoint read. Okay, which means the fluorescence could be a fluorescence contamination or something. It's very easy to see it here. So we don't really get positive NTCs, and particularly not for the valid prime assay. The valid prime assay is one of the best optimized assays we have you don't get a signal on an NTC not even below 45. So we actually use a very good assay here as well.
Pardon me. Am I correct in understanding that you calibrated your genomic DNA using a human NIST standard?
Yes.
Do you have any suggestions for proceeding with this approach for targets for which there are no NIST standards?
I mean, what we do, the thing is, the NIST standard is, first of all, the NIST standard is quite expensive. So what we actually did was that we made a secondary standard. So we make our large batch of genomic DNA that we calibrate against the NIST to get the cost down. That's very, very important. And actually, what we then did was that although I presented data with only a single valid prime, we actually designed 7 of them for the genomic DNA. And then we can run them on the same genomic DNA, and if they all have the same CQs, we know that they agree with each other, so they get calibrated against each other. And then we can actually use one of them on totally different material, and that will be calibrated. So this is how we do. We start with a NIST, secondary standard, and then we go on. Of course, you do get some error accumulation, but it's not very much. And every time we validate it also with digital PCR. So we spend quite some time on it, but that's the approach we use.
Excellent. Thank you very much.